Okay, so today I'm going to give you concept on monoclonal antibodies. So first of all, what this word basically means, mono means single, as you all know. Single, while clonal means copy. And antibodies are here the proteins, okay. So now these are extracted from the single white blood cell, okay. Extracted from single white blood cell, okay. White blood cells. So, and this is here also known as hybridoma technique, okay. Technique, okay. So now here, what are proteins? What these antibodies and proteins mean? So let me explain over here. So these are basically the, what we can say, okay. So these proteins are basically, these can bind only with a special epitome, okay. These and proteins can bind only with specific epitome. Now you will be thinking that what is epitome? So it is basically the binding site of antigen. What? Binding site of antigen. So let me draw here picture and explain you. So for suppose here we have, there is one antigen. Okay, what is this? Antigen. So what epitome is basically the binding site of antigen. So here these are the binding sites of antigen. So these are known as epitome. So there are many epitomes. I just draw 3 epitomes. So epitome, epitome. So let me give okay. Let me epitome 1, epitome 2, and epitome 3. So now what will happen here? So here we have one plasma cell. What? Plasma cell, okay. So this plasma cell will make the antibodies of epitome 1. For suppose plasma cell is making the antibody, what? Making antibody of epitome 1, okay. So this plasma cell will not make another antibody for the epitome 2. So here we will make another plasma cell, another plasma cell which will make the antibody of epitome 2. Okay, means one plasma cell can only make the antibody of only one epitome, not remaining. And 3, there will be another plasma cell which will make the antibody of epitome 3. So this is how the plasma cell make the antibody of specific epitome. So here we have what? Here we have multiple binding sites. Binding sites. 1, 2, 3. Multiple binding sites are there. Okay. So here multiple antibodies are made. As we have multiple binding sites, so what will make multiple what we can say antibodies are made. So these multiple antibodies are known as polyclonal antibodies. Poly means many. So here many, multiple or many antibodies are made. So this is known as polyclonal antibody. Now these polyclonal antibodies will divide divide mitotically. Mitotically, mitotically the division of plasma cell. Of plasma cell. Okay. Into the daughter cells. Okay. Into daughter cells. So they will these plasma cell mitotically divide means they will one will separate from each other. So here we have many daughter cells. So one here we have one, here we have another one, here we have another one, here we have another one. So this separate one is called the what? Monoclonal antibody. Okay, this was known as polyclonal antibody many, and then they will divide mitotically into the one each daughter cell, so they will known as monoclonal antibodies. Okay, so one more thing, so this concept was basically the concept of monoclonal antibodies was given by two scientists in 1975. Name of those scientists are basically George George Connor and another was G. Milston. Okay, and further on, after that, when another scientist came, the name was Neil K. So they three scientists worked on the concept of monoclonal antibodies, and they got the Nobel Prize in 1984. Got the Nobel Prize. Okay, so now here we have one more thing that we have antigen snake venom. Anti snake venom. Okay, so this is basically the this to make this we use monoclonal. We use monoclonal to detect the diagnosis hepatitis, and there are some treatments like cancer cell, tumor cells. Okay, so here we have to detect the diagnosis and you can say hepatitis, and another cell. This is basically in the cancer cell and tumor cell. Okay, so now let me tell you about the process of monoclonal antibodies, how these bodies and monoclonal antibodies are formed, means artificially how these are formed. So let me discuss. So here we are going to discuss the mechanism of production of monoclonal antibodies. Okay, so we were making this as the artificial. Okay, so let's discuss. Here are some steps to discuss this. Let me write here some steps, then I will tell you each steps in detail. Steps. So first of all, we have antigen. Then we have what we can say insertion in living organism. Okay, and after that we have extraction of plasma cell. Then after the extraction we have fission of those cells, fission of plasma cell you can say. Okay, so these are some steps. Furthermore steps I will tell you later on. So here the example which is given in the to make the artificial monoclonal antibodies, that is rat mice. Okay, so let me draw the mice here. Okay, let the wait. So first we have antigen. So we will insert the antigen in the mice body. What will insert the antigen? Okay, so antigen after that, if when we enter the antigen, so this antigen will activate the immune system. Okay, what this antigen will do will activate the immune system. Okay, after activation, plasma cell make the antibodies in this rat. What after activation, plasma cell will make antibodies. Okay, so these plasma cells have not that much more life. Is life expand. Okay, so this plasma cell have only we can say 7 days, 7 5 to 7 days life expand. After that, these plasmas also die. Plasma. Okay, so let's continue on this topic. What happened after activation? Plasma cell make the antibodies inside the body. Then after 6 to 10 days, 10 days, this rat or mice you can say will die. Okay, so plasma cell have also little bit life about the 7 days. So so here we can put the plasma cell. Okay, so let me explain you one more thing. In the rat, most of means most plus B cells. We need here B cells. Okay, so B cells are present most in the spleen. Spleen, the part in our body, or you can say organ. Okay, spleen. Most of the B cells are present. So what we will do here, we will remove out the spleen from the rat body. Remove spleen from rat body. Okay, then after plasma, after removing this spleen from the body, we will select, we will means what we will do. Okay, so here we have another cell, that is Malan cell. Malan cell. So now this cell is immortal cell. Immortal means have the rapid division. Means this cell divide very rapidly. Immortal. Immortal cell. means this have the uncontrollable division. Un- Uncontrollable division. This cell will divide rapidly. Rapid division, you can say. And we take out this from the bone marrow. From where? From bone marrow. Okay, so till that, here I hope it is clear. Now here third steps is done. Extraction of plasma cell is done. Now we will do the fission of plasma cell. So here we have, let me write here, fission of plasma cell, and one other cell was myeloid cell. And one more thing, myeloid cell have means more life expand. It can not die easily. Life expand more. Okay, so here the fission of plasma cell and myeloid cells. So here the chemical is used. In the fission of plasma and myeloid cell, when chemical is used, that chemical is basically PEG, pig. Okay, is basically polyethylene glycol. Okay, so this chemical is used in the fission of plasma cell and myeloid cell. So hopefully till that it is clear to you. So here, when these both cells will fuse together, like here both cells are fused together. So this is means we can say the fusion of both cells is called genetically modified cell. Okay, or you can say this as the hybridoma doma cell. This is one type of specific white blood cell. Okay, so now what will happen here? Here the mitosis division will happen. Okay, so now here mitosis division will happen. Right here, mitosis division. And this is the rapid division. Okay, sorry for the bad writing. Rapid division. Now let me move to the another page. So what rapid division will happen, and that parent cell will divide into daughter cells. Okay, make means different, make the different copies of itself. Like here, separate copy here, one is separate copy here, and a separate copy like this. Okay, so now this is known as culture. Wait, culture techniques. Okay, culture techniques. Now here we give the medium. What we will give one medium. Medium. Sorry, medium. And that medium is called het medium. What? Het medium. Yeah. Now have I we give here one medium which is known as hat medium to promote the growth growth of fused cell. Okay. So that we get the after promoting the growth of the fused cell we will get the antibodies. Then we will extract them. After extract them, now these are called as after extract them, now these are called as autoimmune disorder. Autoimmune disorder. So one thing I want to explain you over here, hybridoma is the importal cell. Okay, so the means the daughter cell will be made. So these will be also the importal daughter cells, means copies of itself. So hopefully this was today's lecture. You enjoyed. I told you the mechanism of the monoclonal. So this was today's lecture. Hope you enjoyed. Bye bye.